Hi, everyone. My name is Will Bailey-Elkin, and I'm tuning in from Winnipeg, Manitoba. So I just want to welcome everyone to the last session of the day. Before we get going with these presentations, I just want to remind everyone to stick around at the end of this session because we'll be voting on the furry or feathered friends category for the photo competition. So traditionally, this is a poster session, but because we're meeting virtually, I believe the two presenters have more of a sort of a PowerPoint style presentation. For me, this is an exciting session because both of our presenters have committed a significant portion of their careers in research to the study of phosphorus management in organic agriculture in Manitoba. And we all know that this is a very important topic for most prairie organic farms. So because both speakers are going to cover similar areas of research, we wanted to hold all the questions until the end after both speakers have presented. Again, you can either put up your hand in Zoom and then it will get to your questions at the end during the question period, or you can type them in the chat and we'll be able to see them there. So I'd like to first introduce Michelle Carkner. I actually had the pleasure of working alongside Michelle as a master's student at the U of M, and I do believe that I helped out with some of the fun and sort of long days of data collection and harvest for some of the trials she's going to discuss. Michelle Carkner is a PhD student working under the supervision of Dr. Martens. Her current research focuses on the evaluation of different wheat cultivars under low phosphorus organic conditions. Prior to pursuing her PhD, Michelle worked as a research associate with the University of Manitoba, where she coordinated a national wheat, oat, and potato breeding program with participating farmers. Then we have Joanne Thiessen Martens. She is currently in the final months of a PhD program in the Department of Soil Science at the University of Manitoba, studying phosphorus dynamics in organic systems where Struvite is used as a phosphorus source. She has worked in organic research and extension for many years, mainly in the area of soil fertility management. Joanne is originally from a small mixed farm near Austin, Manitoba, and now lives in Winnipeg. So I'd like to first pass it on to Michelle Carkner to provide us with an update on her research. Thank you.
Great, thank you, and welcome everybody. It's so exciting to see familiar names. And yeah, what a great turnout. So thank you so much for coming. I'll just share my screen. This one, hopefully. I'm assuming that everyone can see it. So my research is looking at how different wheat cultivars from a participatory plant breeding program differs between each other under low phosphorus conditions. And so the title of my talk is, can we improve the performance under low phosphorus organic conditions by diversifying the selection environment and the selector? So I wanted to kind of jump right into the soil phosphorus cycle. And I apologize if this is a lot of information at the end of a long day. However, I think it's important just to establish the different phosphorus pools within the soil, because the plants are going to interact with these different pools depending on what they are. So plants can really only access phosphorus when they're in this anion form, the soil solution phosphorus. And there's two other pools within the soil that we need to be aware of. There's the inorganic phosphorus. So on prairie soils, we have a lot of inorganic phosphorus, but oftentimes they're tightly bound to mineral complexes, so calcium or magnesium. And then there's the organic phosphorus pool. So this is the phosphorus that's kind of tied up in our organic matter. And it's oftentimes very difficult for the plant to take up because it's in these ring structures. And so we'll talk a little bit more about that. Another way that we can look at different phosphorus pools and accessing them is to look at how these different pools interact with each other. So sometimes there's phosphorus transfer between the fixed pool, so the very tightly bound phosphorus in the soil, and the active phosphorus pool, the phosphorus that's in organic matter. And this transfer happens through microbial activity. And then another way that the active phosphorus pool actually moves into the soil solution pool, so the phosphorus that plants can actually take up is through the decomposition of this microbial biomass. And so these ring structures being broken down through decomposition, sometimes these phosphorus ions can be released and the plants can take it up. And a lot of times, like stabilized organic matter that's very physically protected from degradation can actually be placed into the fixed phosphorus pool, because it's very, very hard for plants and microorganisms to access that. So in accordance to the plant, how do plants respond to low phosphorus environments? So we have all these different pools. We have the fixed phosphorus pool, the active pool, and the solution pool. And phosphorus is very immobile in the soil. And so oftentimes, even if we do have phosphorus that can be actively taken up, plants can actually change their root structures such that they're exploring a higher surface area of soil than maybe some other cultivars. So for example, this is a common bean. You have some roots that can go very deep, some that are standard, and then some that can actually change their root architecture so that they're foraging more topsoil. And so they're trying to increase the amount of surface area and increase their ability to access those phosphorus that is able to take up. Another way that plants can explore more surface area of the soil is by making associations with arbusculomycorrhizae fungi. I'm assuming a lot of you are familiar with AMF, but for those of you who aren't, AMF is essentially a fungus that infects the inside of a plant root. And the hyphae of the fungi can increase the surface area of the soil that it's able to explore. So this green is the mycorrhizae hyphae, and then this is like a root hair. So the mycorrhizae are able to access higher amounts of phosphorus in the soil, just pure physical exploration. And the fungi gives the plant the phosphorus, and the plant gives the fungus carbon. And then another way is soil exploitation. So trying to take up as much phosphorus in the situation that the plant is in. So some plants can actually exude, some plants can actually exude some acids that can break apart these different complexes within the fixed phosphorus pool. And then some plants can actually exude phosphatases, which is an enzyme that can break apart those ring structures in the organic phosphorus pool that we talked about before. So these are different ways that plants can actually access phosphorus when it's very low in the soil. So in organic situations where we aren't adding fertilizers, and a lot of times manure is very difficult to come by, crops grown under organic conditions and oftentimes low phosphorus conditions must place energy into biologically mediated nutrient supplies. So these plants have to interact with that organic phosphorus pool more so than conventional plants, plants grown under conventional production. So how do we measure this? Well, we're going to look at 3 different calculations today from my research. There's phosphorus uptake efficiency. So this is the ratio of phosphorus taken up per plant, per phosphorus available. There's phosphorus utilization efficiency. So this is the amount of grain phosphorus per total P taken. Essentially, with the phosphorus that it was able to take up, how much was actually moved into the grain. And then a combination of these 2 calculations is the actual, umbrella phosphorus use efficiency. So how efficient was the crop at using the phosphorus that it had? Switching gears, because I think this is also a very useful portion of my project, is to explain a little bit about the participatory wheat breeding program. So essentially, it's a program that started in 2011 with Manitoba farmers and expanded Canada-wide in 2013. And it's a collaboration between breeders, farmers, and researchers. The breeders made crosses. We sent those early generation seeds out to farmers, and the farmers planted these seeds on their farms. So this is an example of a farm in Prince Edward Island, and the farmer in the background. And the farmer actually selected from this population. And so essentially they became the breeders. So we have a diversity of farmer breeders, and we also have a diversity of the actual areas in which these lines these cultivars that I'm going to be talking about were selected from. And this is just to give you a clear idea of what the PPP program was and how it differs from a normal breeding program. The majority of the parents crossed and selection and all of these things happen on the research station, whereas in the PPP program, the farmers selected the parents with the researchers. The populations were grown on the farm. The selections were made by the farmer. And some of the trials that we are doing to assess the performance of these lines were done on farmers' fields, but also at the University of Manitoba. So trying to tie this in together with the phosphorus question, it's well understood that the selection environment impacts the final production performance. So this is exemplified through regional breeding programs we have in Canada. So we don't have just one wheat breeding program for all of Canada because of regional differences in environment. And it's also been shown that breeding spring wheat under organic and conventional conditions and then tested under organic conditions have shown that organic produced lines performed better in organic versus conventional. And this research was actually done by Ann Kirk, who I saw in the audience. So if you have any questions about this, maybe you can ask her. And then there's also some work looking at durum wheat lines that were selected under zero till and conventional till and then tested under these environments. And that the zero till selected lines actually had better early vigor in all of the environments. So selection environment matters. So putting this all together, we need crops to function under lower phosphorus conditions and organic conditions. Plants bred under low phosphorus or manure applied systems. We have the cultivars, the genetic material to test this. And we know that there is an importance of the selection environment in a breeding program. So the hypothesis for this research was that by selecting under organic production environments, farmers will select for lines that utilize phosphorus more efficiently and access greater organic pools of phosphorus than their parents under organic conditions. I know it's a lot of information, but yeah, just want to give a little bit of background, and hopefully the phosphorus pool background will also help in Joanne's presentation. So the experimental design, the research is going to be conducted over 3 years. So 2020 was my first, 2021 is my second, and 2022 would be my third. The phosphorus, the soil was very, very low in phosphorus, and the previous crop was a cover crop mixture. It was grown all under organic management. We had very little breed pressure, and we wanted to test 2 different factors. So we wanted to test different genotypes, and we wanted to test different soil phosphorus status, so with manure and without manure. And so these lines, the lines that we tested are from the PPP program. And so PWA00JG is from a line from Ag CBC. PWA00KB is a line from Carmen, Manitoba. These are the parents. So Red Fife, which is a landrace 5602HR, which is a modern variety. These 2 were crossed, as shown in this little illustration down here. The parents were crossed. The F3s were sent to the farmers, and now we have 2 completely different lines, but with the same parents. And so then we randomized that within an experiment. I'm not going to talk too much about the physical mixture, parental treatment, or the F3s. That was to try to answer another hypothesis in my PhD project. We're going to mainly focus on the farmer lines and the parental lines in this research, or in this presentation. I wanted to give a seasonal snapshot through performance. So the research was done in Lebo, which is about an hour or 40 minutes northeast of Winnipeg. And it was very dry in 2020, and then it was extremely dry in 2021. So that's kind of shown here. The growing degree days were very high. Precipitation was low, and in 2021 it was even lower. So I wanted to show this little graph here because this impacts some of the phosphorus dynamics that we saw that we're going to be talking about. So in Lebo 2020, the lines responded more to manure than the lines did in 2021. And that's because it just didn't rain. And so it was a combination of poor mineralization of the manure and also just poor performance on the cultivar's part. So let's just jump right into the phosphorus uptake efficiency between these. So this is the total plant phosphorus per the total available phosphorus. So this is the existing soil phosphorus and the fertilizer phosphorus. And so I've characterized 2020 as being dry conditions, and I've characterized 2021 as having drought conditions. And so this is with phosphorus added. And you can see that the farmer, we didn't have any significant differences between the lines, but we can still talk about the dynamics that have gone on here. So the farmer lines actually didn't take up as much phosphorus than the parental lines did, and red fife was was variable. However, under low phosphorus conditions, so this is absolutely no manure added, the farmer lines seemed to be much more efficient with taking up the phosphorus that was there than was able to take up the phosphorus that was there rather than the modern parental line. And then in the drought conditions, I didn't really see any general pattern here. And so even under drought conditions with low phosphorus, you can see that if you look on the axes here, they just weren't able to take up as much phosphorus as per what was there compared to slightly moister conditions. So moving into phosphorus utilization efficiency. So as a reminder, this is how much phosphorus they were able to take up in the whole plant and how much they were able to move into the grain. And so under dry conditions in 2020 with manure added, 5602HR seemed to move more phosphorus from the biomass of the plant into the grain as compared to the farmer lines. And then in the low phosphorus conditions, it seems like PWA00KB translocated more phosphorus, moved more phosphorus from the biomass into the grain compared to the modern cultivar and one of the parents, and it was very similar to red fife. And then under drought conditions, it looks like red fife translocated less than the other lines, and PWA-JG moved less. And then in the drought, low phosphorus conditions, there isn't too much of a pattern there that I can talk about. So combining these together, so this is uptake efficiency and utilization efficiency combined together to represent the phosphorus use efficiency. So what is the contribution supplied from soil P to the final yield phosphorus, the amount of phosphorus in the grain? And under added manure conditions, 5602HR took up more phosphorus and moved it into the grain much more than the farmer lines, whereas in very, very low P conditions, the farmer lines did a better job. And then in the drought conditions, there didn't seem to be too much patterns that I can talk about. And it's probably because in general, the plants were not performing very well and they weren't able to take up the phosphorus that was even there due to the very, very poor drought conditions. So why do we care? So was phosphorus use efficiency driving yield? How does this all relate to the final production? Well, it's interesting because it really depends. Under very low phosphorus conditions, when I did some correlation analysis, we did have a significant relationship between phosphorus use efficiency and yield under low phosphorus conditions. However, under higher, the manure added conditions, there was no significant relationship between yield and phosphorus use efficiency when there was adequate phosphorus available. Whereas in the drought year, we had a significant relationship at the low phosphorus level, and we also had a significant relationship at the high phosphorus level. And my guess is probably because these lines were kind of acting like they didn't have enough phosphorus, and it's probably because they didn't have enough phosphorus because it was so dry and they just weren't able to access phosphorus. So I think this is really interesting because under low phosphorus conditions, how the plant takes up and uses that phosphorus really impacts final yield, whereas when there's adequate phosphorus, the use efficiency of the plant doesn't seem to matter as much. This is all very preliminary. So a little bit of discussion points. We didn't see any cultivar differences between the treatments and efficiency, but that doesn't mean we can't talk about the general patterns that we did observe. However, under low phosphorus conditions, farmers seem to take up more phosphorus than the modern parent, and the farmer lines seem to be more efficient under low phosphorus conditions. And 5602 responded more to added nutrients than the farmer lines, which is interesting. However, thus far, the efficiency and the uptake seem to be more related to environmental conditions more so than the genetic differences or the selection environment these were selected under. But I think a lot of that has to do with the poor moisture, because a lot of other researchers have found significant genetic differences in their research. So I still have confidence that 2022 is going to be good. So one question to ponder is that a lot of the phosphorus use efficiency calculations that I see in papers that people talk about are actually derived from nitrogen use efficiency, but not really sure if this is appropriate, because moving more nitrogen from the biomass into the grain is useful, because then you get higher protein content and that increases marketability nutrition. But that's not necessarily true for phosphorus. It's actually a detriment to have more phosphorus in terms of nutrition, because our bodies and a lot of other monogastric animals can't actually break down the phytate that's how phosphorus is stored in the grain. So this idea of phosphorus utilization efficiency, moving more phosphorus into the grain, it doesn't seem to be connected to yield. And so this might be a great opportunity to examine cultivars with lower phosphorus utilization efficiency and also high yield under organic conditions. So what is the ideal wheat plant under organic conditions? Possibly we could take a plant that can take up phosphorus and use it to make biomass and move as little phosphorus into the grain and leave phosphorus on the farm. But there are obviously implications of this. So for example, like early vigor, if you save that seed, early vigor in the next season. So yeah, these are some questions to ponder. But that's the end of my presentation. And I'd like to thank my advisor and technical support. And that's Will on the Hay. So yes, thank you very much. And I guess I'll just move it on to Joanne.
Thanks, Michelle. That was really interesting, and I'm just make sure you can hear me and see my slides. Yes, great. So yeah, Michelle and I have actually worked together quite a bit when we were both working with Martin Ants, and it's pretty neat to see how we're both advancing our understanding of phosphorus coming out of that low phosphorus environment that we had in the Ants team research. So my work that I've been doing in the last 3 years or so as part of my PhD is looking at struvite as a possible phosphorus source for prairie organic crops. And we'll talk a bit more about struvite and how it potentially fits into organic cropping systems in a minute, but as some of you probably know, this isn't really an option that's completely available to you right now. And so another way to, for viewers to think about this presentation, another lens to look at all of this through is just in general, what happens when we add phosphorus of any kind to these really low phosphorus soils with different types of crops and that kind of thing. Struvite's a bit unique compared to some other phosphorus sources for organic, but a lot of the lessons that we're seeing from the struvite work can be generalized to other phosphorus sources as well. So let's talk a little bit about phosphorus management in organic agriculture. And I'm kind of preaching to the choir here a bit, but I think it's good to review a few basics. So soil phosphorus deficiencies are very common on organic farms, and that's been documented all over the prairies with these, very low, 3 parts per million, 4 parts per million Olsen phosphorus. And often that's not due to any sort of poor intent on the part of the farmers, but often it's due to just a lack of suitable phosphorus inputs. So, so that's a problem that's really hard to face, and that's part of the reason why I'm looking at Struvite in the hopes of adding that to a practical and available source that we can use in organic. Then the other thing to think about in our phosphorus management in general, not just organic, is that in agriculture we have this open phosphorus cycle where very little of the phosphorus that is, mined in Morocco, for example, or other places, and then transported all over the world and fed to crops, and then those crops fed to animals or to us as humans, very little of that phosphorus actually gets recycled back to the land. And that's a problem, because phosphorus is a non-renewable resource, and when the mined sources run out, then that's it. So we need to be thinking about recycling as much as possible, and the principles of organic actually really support this. They emphasize recycling and efficient management of all kinds of materials and energy. So let's think then about, in general, what makes a phosphorus source suitable for organic. And this is something that I'm going to apply to Struvite, but we can think about for any kind of input that we might be using. So some key things that we want to look at are, of course, do we get a good crop response? That's pretty obvious. But there's a lot of other factors as well that we want to consider. We want to have a low level of contaminants so that we're not loading up our soils with heavy metals or moving pathogens around and that kind of thing. Ideally, it's recycled for the reasons we just talked about. We want it to be at least relatively easy to use and cost effective. And then clearly it has to be available, as in we can get our hands on it and actually use it. And then this is a bit of a nebulous one, but we wanted to have kind of a moderate solubility to be good for organic systems. And that has to do with a couple different things. One is that if we have a phosphorus product that has very low solubility, we're not going to get any crop response. So clearly we don't want a very low solubility, but we also don't want a very high solubility, because those really soluble fertilizers do have an impact on soil organisms, in particular the mycorrhizal fungi that Michelle was talking about, and earlier Mario Tenuta was talking about these, and how we kind of need that sweet spot for the mycorrhizal fungi to really work well. I was looking into this a while ago, and it seems that plants actually need to experience a little bit of phosphorus deficiency early in their life to initiate that relationship with the mycorrhizal fungi. So they need to sort of feel that pinch a little bit. But then if the mycorrhizal fungi doesn't actually manage to find phosphorus out there and provide it back to the plant, then the plant will cut off the relationship. So it's another angle to think about with that sweet spot is, the plants need to not have super easy access to phosphorus, but they do need to have some access. So that's why that moderate solubility might be important. So let's think about Struvite, give you a bit of background on it, and then think about how it might fit in as an input for organic systems. So Struvite is a mineral. It's made of magnesium, ammonium, and phosphate, and then it has these high water molecules all put together in this crystalline structure. And it's a naturally occurring mineral. It naturally builds up in the pipes of wastewater treatment plants like this. And so because all those nutrients are there and the conditions are right, it'll naturally precipitate there. So the wastewater treatment folks are anxious to have this not happen in their pipes. And so they've been looking at ways to make it happen elsewhere in the process to help keep the pipes clear. And then when you do that, you get this kind of this white granular sort of fertilizer product that's rich in, especially in phosphorus, and then with a bit of nitrogen and some magnesium in it as well. It has quite poor solubility in water. It's less than 5%. And but in acidic conditions, if you apply a citric acid to it, then about half to all of it will dissolve under those conditions, depending on the purity of the struvite and that kind of thing. So there's been a bunch of research done, mostly in greenhouse settings, like in pot experiments, a few field experiments, looking at how crops respond to struvite as a fertilizer. Most of them do find that it is better than rock phosphate. It sometimes can perform similarly to soluble fertilizers like 1152 or some of the other fertilizers we have available. It tends to respond, crops tend to respond not as well in high pH soils as they do in neutral or acidic soils. The problem is not nearly as bad as it is with rock phosphate, but there is a tendency for it not to work as well in alkaline soils. But then some of the interesting things are that there's some differences among crop species that show up. Like certain crops seem to be able to use struvite really well and others not so much. And so it emphasizes the importance of some of those processes that Michelle was talking about, those mechanisms that plants use to actually get phosphorus out of the soil. They can use those same mechanisms to get phosphorus out of a fertilizer as well. And so there's a lot of speculation there about what's happening, but not a lot that's been actually shown. But what all of this research has not really done to this point is look at how we're going to manage this stuff if we're using it. Like, so what's the appropriate application rate? Where should we place it in the soil, like banded or below the seed, with all those kinds of things, timing of application. So there's very little agronomic work that's been done. So with that in mind, then let's think about whether struvite helps or can meet some of these criteria that we're looking for a good phosphorus source. It does actually have a very low level of contaminants due to the way it's extracted from wastewater. Because it's a crystal, it just forms with those different elements that are present in the wastewater without actually bringing any of the other stuff along with it, or at least very, very low levels. So that's a good start. It is recycled because they are recycling this out of human waste, municipal wastewater, and it can also be extracted out of things like out of manure or out of, say, food processing waste, other non-human sources. It's easy to use because it's in a granular format, so you can just put it through a fertilizer box or whatever, your air seeder, and it's pretty cost effective compared to a lot of the other organic inputs. It's more than the conventional soluble fertilizers, but not a whole lot more. So it looks really promising that way. The big downfall for struvite right now is that it's not functionally available for organic production at this point. In the last revision to the organic standards, struvite sourced from non-human sources was added to the permitted substances list. However, I don't actually know of any sources that are commercially available of that type of struvite. If someone else knows, let me know, and I can guarantee there's a lot of people that would be interested in hearing about that. The human sourced struvite is commercially available. That's the fertilizer Crystal Green. You can buy it here and use it, and some farmers already are. But that is not permitted in organic in Canada at this point. And hopefully that will change in the future. Now, the solubility issue. So, I mean, when you do tests on this stuff in the lab, it is sort of a moderate solubility, but we don't know a lot yet about the crop response part, and even less about how the AMF, the mycorrhizal fungi, respond to it. So I did a couple of field studies looking at some of these questions. I'm going to focus on this first study where I did try to get at some of those management questions, specifically what's the appropriate application rate of Struvite for some different organic crops. So I wanted to look at, what's the crop productivity and the phosphorus uptake of these crops, because clearly we want something that will actually boost our yields. But then also to look at, what are the effects on the soil phosphorus pool that's left behind, because we do want to boost that up if it's really low, but we don't want it to be too high because that starts to pose a risk of environmental losses. And we also I wanted to see what the effect would be on the mycorrhizal colonization of crops. So in other words, trying to optimize these three things, so what Struvite application rate will support good crop yields without suppressing mycorrhizal fungi like a lot of soluble fertilizers do, or raising soil P too much. So I looked at flax, and I looked at spring wheat, and I looked at an alfalfa grass forage. So I did this research at Lebo, Manitoba, the same site that Michelle was using. This was just a little before she was doing it. It was in 2017 through 2019. Also dry years, but not as dry as the last couple that we've had. The soil there is an alkaline pH, so it is a challenging environment to test Struvite in, but that's what we wanted. And it is a very low phosphorus concentration in that soil. So we did the experiments with wheat and flax by basically repeating the same experiment 3 times in the 3 different years on different locations and applying 3 different rates of Struvite. So either 20, 30, or 40 kilograms of actual P per hectare. So in P205, that's, multiply those by 2.3 or something like that. So, we're up around like 45, up to about 90 kilograms per hectare of P205. And we banded it with the seed in the wheat and flax experiments. For the alfalfa grass forage, we took a bit of a different approach. And there we again used 3 different rates. They were a little higher this time because we were taking a multi-year approach. So we applied the Struvite in the spring of 2017 and then didn't apply any more, but just kept monitoring those plots over the next 3 years. And we banded it into an existing forage stand that was really depleted, was performing very poorly, just to see if we could rejuvenate that forage stand with the Struvite. So let's start with the alfalfa grass. I'll walk you through this graph. Basically, we did find that the increasing application rates, which are shown on the x-axis here, did increase the yield of the forage. And the interesting thing is that the effect got stronger over time. So the blue line here is the relationship that we saw in the first year of the study, so in 2017. So on the left, that's the control where none was applied, and then the yield gradually increased with the higher rates in the first year of the study. Then in the second year of the study, even without adding any more Struvite, we got a stronger response. So it really seemed like that Struvite was living up to its sort of slow release reputation and was continuing to release more phosphorus to the crop. Either that or the crop was just able to access more phosphorus because maybe its root system grew bigger in the year before or something like that. Whatever the reason, we got a stronger effect in the second year than in the first. And then in the third year, we also had a fairly strong response. So it was a nice long-term effect that we saw from adding a Struvite and a one-time application at the beginning of that experiment. The tissue phosphorus concentration also increased. I didn't show that data, but we saw it increasing as well, which is a good sign that the plants were actually taking up a lot more phosphorus when we added the Struvite. Now, one thing you'll notice is that these lines don't really level off even when we get to the high rates. And so we don't actually know what the maximum yield would be that we could achieve with the Struvite here. Perhaps it's even higher than this 90 kilograms per hectare. which is important to keep in mind for management. The 30 kilograms per hectare rate was actually the recommended rate for that soil type and crop. And so we had to triple that rate to get those really high yields. We'll talk more a bit about that in a minute. The other interesting thing about the forage, the alfalfa grass forage, is that in this mixture with grasses, there's timothy and smooth brome or meadow brome. It was only really the alfalfa that responded to the struvite. On this graph, the alfalfa part of the forage mix is shown in the dark green, and then the gray is grasses, and the little red slice is the weeds. So you can see the gray and the red are pretty much the same across all the different application rates, whereas the alfalfa chunk, the dark green one, is the one that really changes with the increasing rates. So the control treatment was dominated by grasses, very little alfalfa, and all the other ones were dominated by alfalfa to varying degrees. So this is really important in general, showing the role that phosphorus plays in our forages, because this has huge implications for not only the forage quality, the protein content of that forage, but also if you're using alfalfa in your rotation to fix nitrogen for future crops, and you only have this much alfalfa, that tiny amount in your mix, you're not going to have a lot of nitrogen available. So phosphorus, adding phosphorus to those forages is really important for supporting the nitrogen supply to those next crops. Now, when we look in the alfalfa stand, look at the mycorrhizal fungi and the soil phosphorus. There's some interesting things going on, interesting in that not a lot is happening. So even though we applied all that phosphorus to this, we didn't have a significant effect on the mycorrhizal colonization in the alfalfa roots. Here you can see those little blue sort of branched little structures. That's where the phosphorus and carbon exchange is happening inside the plant cells. And they were not really affected by the addition of struvite, which is really promising. The soil phosphorus levels, there was not really a significant effect by the kind of the traditional measure of what's significant, but there is an increasing trend where soil phosphorus increases with application rate. But you see, even our highest one is only just below 10 parts per million, Olsen P, which is a very reasonable Olsen phosphorus concentration in your soil. It's certainly not creating risk of environmental losses really at that point. So again, that looks pretty promising. Let's talk about wheat now for a minute. So here, the graph with the green bars shows the yield with the different rates on the x-axis. You'll see we again had that sort of gradual increase with the increasing rates. Not as dramatic as with the alfalfa, but it was there and it was significant. But at the recommended rate of 20 kilograms per hectare of P, there wasn't much of an increase. It took higher rates to actually get those increases. With the mycorrhizal fungi, we did have a little bit of suppression of the fungi at the highest application rate, but it wasn't actually significantly different from the control or from the third one. It was just lower than the recommended rate. So a little bit of something happening there. No effect at all on the soil phosphorus levels in the wheat experiment. In the flax, nothing at all was significant. The yield was completely flat. The mycorrhizal fungi was basically completely flat, no effect. Soil phosphorus also flat. Flax is not responsive to phosphorus fertilizers in general to some degree, and so it's not a big surprise that it didn't really respond here. We think that, these were decent flax yields, and so we think that the flax were just relying heavily on those mycorrhizal fungi, and didn't need any additional phosphorus from the struvite in this case. So summing up those field studies, we can see that we got a really strong response from the alfalfa, especially in those second and third years. We got a moderate response from the wheat, nothing really from the flax, but that's okay. In general, we needed really high rates to achieve those yield increases. But the interesting thing is there was lots of struvite left over for future crops to use, like we saw in the second and third years of the alfalfa. And in the wheat and flax experiments, even in the spring of the following year, you can see in this photo on the bottom right, we could find partially dissolved struvite granules in the soil. So that struvite was just slowly dissolving and releasing the phosphorus over time. So even though we needed a high rate in that year, it could be that that rate would be enough to supply several years worth of crops. We saw not very much effect on mycorrhizal fungi, except a little bit in the wheat. And we didn't see any concerning effects on soil phosphorus. So overall, from this study, it does seem that struvite is pretty promising as a phosphorus source. I want to tell you just a little bit about my other field study, just really briefly. I wanted to see then, if some crops respond well to struvite and others not so much, like what is it about those crops that makes this happen? So why do only some plants respond well to struvite? And this goes back to some of those mechanisms that Michelle was talking about. So acidifying the rhizosphere or producing these, they're sometimes called organic acids or organic anions like citric acid, which despite their name, they don't actually really acidify, but they have a different mechanism of really helping to release phosphorus from the soil. So I did a field study to look at a few different green manure crops, field pea and faba bean and buckwheat. Buckwheat has been shown to respond well to struvite in a greenhouse experiment in the past. And so I thought that was a good candidate. And these others are good green manures for our region. So I want to see if these crops can respond well to struvite, and if so, maybe why is that? So I did a field study at 2 sites, again at Lebo, and this time at Notre Dame, or the tree kern area. Both alkaline soils again, but this time the Lebo soil is very low in phosphorus, and the Notre Dame soil had a moderate phosphorus availability. And so we applied struvite, or we applied monoammonium phosphate, 1152, in 2020, or to a spring wheat crop grown in the following year, but I'm not going to talk about that much part because we don't have that data back yet. So looking at how the green manures responded, we only got a crop response at Lebo, at the low phosphorus site, and actually only got a response from one of the crops. It was only from buckwheat. It's the only one that where the fertilized green manure actually out-yielded the unfertilized one. So here you can see the green bar is the struvite, and the orange bar is the one fertilized with 1152, and with buckwheat, those were statistically the same, and they were greater than the control. In all other cases, there was no difference from the control. And you can see from the photo here, this is on the far right, that's what the buckwheat looked like without any fertilizer added. And then the struvite and the map looked very similar and showed really nice growth. So then I thought, well, buckwheat must be acidifying the rhizosphere and doing all these other things. When I looked at the rhizosphere pH, so that's the pH of the soil from right around the roots captured in the solution, then we saw some interesting things, but not really what I was expecting. So the black bar is the pH in the soil that had no plants growing in it. And then blue is the buckwheat, orange is the peas, and green is the faba beans. So the peas and the faba beans actually managed to reduce the pH by about 0.6 to 0.8 pH units right around their roots. And the buckwheat, there was maybe a bit of a trend, but it wasn't significant. So it's a bit counterintuitive in terms of what's going on with these different crops. So it's still a bit of a head scratcher and working to figure that out, and it'll probably take more research to really answer this question. But so far we've found that, you know, the one crop that actually responded well to Struvite at Lebo is also the only crop that didn't actually really acidify the rhizosphere significantly. So we'll work on that question a little bit more, maybe in the coming years. It was interesting to see, though, the amount of acidification was the same for all the different phosphorus treatments. So it doesn't matter if they were fertilized or not, or with what fertilizer, they acidified the same amount. And I'm still working on the data from the organic anions. So we're running out of time here, but I just want to say that basically Struvite does seem to be a good phosphorus source for some crops. It does seem to do reasonably well at supporting the soil microorganisms as well. But there's still a lot of questions about how to manage this well, how plants access it. Long-term effects are going to be really interesting, and then looking at blends with other inputs to perhaps get, you know, the best of all worlds in supplying phosphorus to different types of crops. So thank you very much for your attention, and we'll hope to take some questions.